So again, thank you very much for coming. And I now just want to give a very brief introduction to Leslie Gunatilaka. I know Leslie because I used to work for him for about 2.5 years at the Natural Product Center. And if you read the piece of publicity that I put together, you will have noticed that I said that the NPC is one of the jewels, undiscovered, in my opinion, and certainly unpublicized jewels of the University of Arizona, where, and I used to memorize this mantra, where we grow exotic and desert plants under hydroponic and aeroponic conditions. We then extract compounds from them, we characterize them chemically, and then we test them biologically to see whether or not the compounds, the metabolites produced by the plants and the associated fungi have any potential for being medicines against a variety of issues. And I think this ties very well with the movie that some of you just saw a few minutes ago, because basically this is what Dr. Gunatilaka is doing. He's leading the charge on this progression of events, putting traditional medicine from China, from Native Americans, from India, from Sri Lanka, all to the test of science to produce new medications. So with this long introduction, I want to welcome Leslie and hope that you will have a good time talking to us about new discoveries. Thank you for coming.
Thank you, Rafi. Can you hear me well? Can everyone hear me well? Thanks, Rafi, for those very kind words of introduction. I would also like to thank Professor Gruner for inviting me over here and giving me this opportunity to share with you some of our recent work on plants, the molecules, cool molecules from plants and microorganisms of the Sonora Desert. Actually, my story begins around the time I visited about 20 years ago for my interview, my current position interview at the Office of Arid Land Studies. And that was actually, it was a surprise to me because I originally come from Sri Lanka, which is a green country, it's green all over. Then I was in Virginia, at Virginia Tech, for about 8 years. Again, except during winter, it's green all around the year. And I come here, while the plane was landing, I noticed it's all brown. So it's the first time I visited the desert. And then, some questions came to my mind. How do some plants survive? Because some of them appear to be green. How do they survive hot Arizona desert? Or how do they survive heat stress? And also, I wanted, as a chemist, I'm trained as a chemist, I wanted to know whether there are any cool molecules, interesting molecules produced by these plants, and also not only plants, and also microorganisms, which live in association with plants, very close association. Are they helping the plants to overcome these stresses met by the plants because of the desert environment? Now, next thing is, since I was going to join the College of Agriculture, trained as a chemist, I wanted to find whether there are any molecules which will have agricultural and also medicinal applications. So that was my main question, which worked. I didn't know whether I did my interview well because all the time I was thinking about these questions. So then I looked at the temperature variation in Tucson. Now this was done recently, 2013. 2013, you see the average temperature variation in Tucson is about 20 degrees Fahrenheit. And on March 2nd, the difference was 75 degrees Fahrenheit. And how, in our case, what we do is, when it is hot during summer, we grab something from a restaurant and we run to an air-conditioned place. We are mobile. Whereas plants are immobile. They have to stand there and fight against this harsh environment. And how do they do that? And they do that, by producing a variety of molecules. And my hypothesis, when I joined, actually I submitted a proposal to the NIH. The review committee liked my idea, my hypothesis. My hypothesis was that survival, that is damage control, of plants with no morphological adaptations. We know that a lot of desert plants have morphological adaptations, but some of them don't have morphological adaptations, or they have little morphological adaptations, and they may require physiological conditions mediated by small molecule natural products, which are capable of modulating heat shock response. That is, they can activate heat shock proteins. Now, I don't know whether you are familiar with heat shock proteins, but I'm sure you have experienced it when you are trying to fry an egg, right? You find that up to a certain temperature, eggs in a pan will remain not doing anything. Then suddenly, everything will become a solid. The reason is it has reached a temperature, stress point, where the proteins will start aggregating. And that is why you end up with this, like a scrambled egg. So now what happens during this heat? What happens is you get, due to heat, proteins in all living organisms undergo denaturation. And this denaturation can be unfolding of proteins, aggregation of proteins, or degradation. In worst case scenario, they can undergo degradation. So what happens? They lose their activity. So as a result of, if there is no activation of heat shock response, or they are also called chaperones, guardian proteins, they take care of other proteins. These are required to maintain integrity of cellular proteins and functions. Now, up to a point, they can take care of these, but after a certain point, they can't. So therefore, heat shock induction or heat shock protein activation is very important for living organisms. Now, what is the connection between heat shock response for agriculture and medicine? Now, in the case of plants, as I mentioned earlier, due to temperature stress, proteins can undergo denaturation. So you need heat shock modulation. If there is no response, then they will die. Right? They can't withstand very high temperature. So therefore, in order for them to survive, you have to upregulate heat shock proteins. So the heat shock proteins will be active and will take care of those damages as a result of these stress conditions. Then in the case of neurodegenerative diseases, most of the neurodegenerative diseases are due to protein aggregation in the brain. Right? Both Alzheimer's and Parkinson's diseases are due to that. And here also, there are the proteins which are undergoing denaturation or aggregation. As a result of denaturation, they aggregate. So again, you need upregulation of proteins. So that is a connection between plants and neurodegenerative diseases. Then when you go to cancer, cancers, as some of you may know, they are highly stressed. They are highly stressed because they are dividing very fast. They need a lot of nutrition, and they are starved all the time. And also, cancer tissue is highly acidic. So therefore, they are always under stressful conditions. So therefore, in the case of cancer, if you want to cure cancer, you have to deregulate. You have to downregulate heat shock proteins. Because it will cause selective cytotoxicity. So for plants to survive, you need upregulation of heat shock proteins. For neurodegenerative disease, you need upregulation. And for cancer, to treat them, you need downregulation. Normal cells do not. They have heat shock proteins, but they are not dependent on heat shock proteins for their survival. So therefore, targeting heat shock proteins for cancer drug discovery is very important because it will cure cancer without affecting normal cells. So that causes what you call selective cytotoxicity only to cancer cells but not to normal cells. So I will just digress a little bit, change my gears and talk about our drug discovery program, or rather natural product discovery program at the Natural Product Center. So we have actually a collection of a large number of plants collected in Sonoran Desert, about 5,000 plant extracts, and fungal extracts. We have about, these are plant-associated fungi. Fungi which live inside plants and outside in the rhizosphere. So they live in association with each other, and we have about 4,000 of those, and we have produced about 20,000 fractions from these extracts. So what we do is, in addition to our in-house assays, we give it to our collaborators. I'll show you a list of collaborators we are having right now, and then we subject these to biological assays, in-house biological assays for cytotoxicity, to see whether any of these extracts kill cancer cells selectively without killing normal cells. We also have two other assays for cell migration. I'm not going to talk much about that today. And also a very important assay, which is the heat shock induction assay, which I mentioned a couple of minutes ago. Now here, we use reporter cells transfected with green fluorescence protein. So what happens is, if there is heat shock induction caused by one of these extracts, it will express green fluorescence protein. So you can see it very clearly under a microscope, and sometimes you can even quantify it. And then what we do is, this is a very laborious process. Rafi mentioned this. We carry out what we call a bioactivity guided fractionation, looking for only biologically active compounds. A plant or a microorganism can produce up to 100 small molecules. We may be looking for 1 or 2 very minor small molecules. So it's a very laborious, very time-consuming process, and some people call it, it's like looking for a needle in a haystack. But my guys in my lab are highly experienced, and they work very hard towards this. And then, we isolate what you call biologically active or promising bioactive metabolites. I'm going to talk only about 2 classes of metabolites today. We have isolated a large number. One is monocillin, which I will show you. We have found to have some agricultural applications. That's what we wanted to find. And then a class of compounds called withanolides produced by plants, which have some medicinal applications. So then what we do is, I don't want to go into details of these. These are highly, technical, structure elucidation, evaluation in secondary bioassays, and we also convert particular compound into large number of analogs to see whether we can improve their biological activity and reduce their toxicity. And that is called medicinal chemistry. So at our center, we do many things. We grow plants, we grow microorganisms, we do biological assays, we do bioactivity guided fractionation, and also we carry out medicinal chemistry to prepare derivatives. Only in vivo assays are done by our collaborators, because we have no facilities to carry out animal studies. Animal studies are done by my collaborators. Sometimes we also try to find what the target is. I will just mention one particular target which we found for a compound called withanolides A, which we isolated from ashwagandha, your favorite plant. And then another problem faced by scientists in the past, or scientists who are involved in drug discovery, is the unavailability of sufficient material to carry out animal studies and human trials. Taxol is a very good example. Taxol almost died because of the supply problem. So when we find a molecule, an interesting molecule, we try to carry out in parallel methods to improve the production. We do various, sometimes blindly, sometimes by accident we discover methods, and then, whatever method we use, we try to produce them efficiently. And also, I'll illustrate all these later. And also what we'll try to do is, we try techniques where instead of doing medicinal chemistry, we'll get the plant or the microorganism to produce a variety of compounds. so that they will do the job for us and we will test them. So that's a very efficient technique. And then also we will see whether these molecules have some new applications once we have discovered them. So these are the, we have, drug discovery cannot be done, or natural product discovery cannot be done in a watertight compartment. It is a must that you collaborate with so many people. If you try to work in a watertight compartment, you will never succeed. I know that, right? So we have collaboration with scientists from the University of Arizona, from School of Plant Sciences, Arizona Cancer Center, Chemistry and Biochemistry, College of Pharmacy, and Immunology and Virology. And all these people, now I am mentioning only those people with whom we have had productive collaboration, where we have applied for grants, got grants funded, or we have published together. Otherwise, the list will be too long. So these are national collaborators: Arizona State University, Translational Genomics Institute, Whitehead Institute at MIT, Harvard University, University of Michigan, Josephine Ford Cancer Center, Nuogen Research, which is based in Tucson, DuPont, agricultural company, Valent Biosciences, agricultural company, Genspera, another company involved in drug discovery, 21st Century Therapeutics, NIH Molecular Library, Small Molecule Library, Repository, National Cancer Institute. We are having very productive collaboration with NCI, and then Scripps Research Institute. Then internationally, I have collaboration in Brazil, several places, very productive collaboration in Canada with a scientist from Toronto. Instar Therapeutics, I'll mention a little bit about this company based in Canada, and then China Pharmaceutical University, and of course, University of Sri Lanka, university from my home country where I was trained. First, I will talk about, yes. Sure, yes. Zika virus, no, I'm not involved. Yeah, so that's the only comment I can make. But I hope they will pass some money for cancer immunotherapy, right, and so on, right, or even candidacies, because we are involved in HIV and discovering drugs for drug-resistant Candida albicans. Okay, so these are the symbiotic microorganisms. I mentioned that we are looking at microorganisms which live in association with plants. So these are the symbiotic microorganisms which we have looked at, plant-associated microorganisms. We have looked at actually plants which grow under controlled environment. Rafi was involved in that work when he was at our center. Then we also look at, or mainly we look at organisms which live in association with plants which grow in natural environment, like endophytic, those are the ones which are found inside, and rhizospheric microorganisms. And rhizosphere is a region where my collaborators say it's like an Amazon forest of microorganisms, because rhizosphere is very rich in nutrients and there are millions of microorganisms there. And then the reason why we are looking at this is to isolate bioactive molecules, and also sometimes looking for unique structural types. Now, I had a project funded by the NIH Director's Roadmap Program to produce new molecules which are new to the NIH library. NIH library has, maybe about 50,000, 75,000 molecules. So it is very hard to find something new to that. And we have found about more than 120 molecules which are new to that library. So what they do is we deposit these compounds with them, and they provide it to all the investigators who are interested in finding biological activities of these molecules. So we have done that. And also, as far as bioactive molecules are concerned, we were looking for those which can be useful in agriculture and in medicine. So today I'm going to talk about, as far as microorganisms are concerned, microorganism which was collected from rhizosphere of a plant, endophytic, that is from inside of a plant, and also microorganism which we have been able to culture, which comes as an endophyte from a plant grown under environmental conditions, under aeroponic conditions. Okay, so this is, we, in collaboration with my collaborator at Arizona State University, we have looked at many microorganisms, and two of the interesting ones actually came from what we call the Christmas cactus. So very beautiful cactus which grows in Sonoran Desert, and this particular fungus called Rhizosphere-associated was collected from the rhizosphere of Christmas cactus. And then the second one was collected from Moment tea, Ephedra fasciculata. It's a type of Moment tea, and it was found inside this plant. And that was identified as Chaetomium chiversii. Now both these, we actually used our assay, which is called the heat shock induction assay, to isolate the biologically active compounds. We came across two molecules which are related to each other. You see the difference between these two is this has an additional chlorine in the molecule compared to Monocillin I. Now unfortunately, this was found to be toxic, more toxic to animal cells, mammalian cells, but this was much better. So we decided to pursue that. And we showed that it was active in heat shock induction assay, and also we showed that actually induces heat shock response. And so it was very important, and this was published in 2006. So here's the molecule. Then, yes. Sure. Oh, sorry. This one? Okay, so we wanted to, we found the activity in mammalian cells. We want to see whether it will affect plants, right? Because I mentioned the connection between plants and mammalian systems, right? If it has anti-cancer activity by expressing heat shock proteins, then there's a possibility that this compound, which is capable of expressing heat shock proteins, might impart some thermotolerance, stress tolerance to plants. So we wanted to try that. And so certain questions came to our mind. The first question was, does this compound, Monocillin I, modulate plant heat shock proteins? For example, HSP101, that's a protein. If so, can it confer thermotolerance, that is temperature tolerance, to its host plant? Then the third question we asked was, does the fungus assist the host plant to overcome stress? And the fourth one, what are the implications? Does it provide proof of concept for the use of heat shock inducing natural products to modulate stress tolerance as an alternative to genetic modification? Genetic modification people don't like, but if we can find an alternative way, it will be really nice. So what we did was, I convinced my graduate student, who is now at National Cancer Institute, to get involved in this work because he's a cancer biologist. He said, no, Leslie, I don't want to do this work. He said, but please do it as a favor for me to see whether it has any activity. So what we did was, we grew Arabidopsis, and then we sprayed a solution of Monocillin I. And to our surprise, pleasant surprise, we found that plants survived up to 45 degrees centigrade for 75 minutes. 45 degrees centigrade, that's very high, right? 75 minutes, and they were alive. Now those plants, and we use what you call a hypocotyl elongation assay, those plants which were not sprayed with Monocillin I, they did not survive. They were dead, right? These survived, these were alive. Then we want to see whether there is expression of heat shock protein 101. And we showed that by semi-quantitative PCR. And also, we had access to what you call HSP101 null mutant, where HSP101 gene has been deleted, and there was no activity. So that confirmed that it was HSP101 which was overexpressed by this molecule. So then, next step was to see whether we can really grow them together and get the Arabidopsis plants to survive high temperature. So we did this experiment. We grew plant, seedlings, and also the fungus together in the same medium, and we found that again with the fungus the plant survived. And then what we did was we actually analyzed the medium and found that what the fungus did was it exuded Monocillin I into the medium, and the plant took up that exuded Monocillin I, and it was able to survive high temperature, 75 minutes at 45 degrees centigrade. Then we did actually a soil experiment. Using sterile soil, we added the fungus, our fungus, Rhizosphere quadriceptata, and then without fungus, and also we used a controlled fungus, just a dummy fungus, which had no biological activity at all. We showed that it had no biological activity, and again, under greenhouse conditions, we were able to see this effect. Survive for 2 hours at 45 degrees centigrade, and this was actually DuPont got interested in this, and then they closed down their natural product section. Then Valent Biosciences got interested. That's a subsidiary of DuPont at that time. And then they actually carried out this Monocillin I experiment with crop plants and found that it was effective. But unfortunately, what happened was, by mistake, we did not cover our discovery. Intellectual coverage was not there. We didn't have a patent. So they said they're not interested. So that was dropped by them. Now, a Chinese group, a university is picking that up, and we have just signed agreements to send our microorganisms to them to study, because there's no future for it in the US, because we have not covered it by patent. This was published in Plant Physiology. It was highlighted in Science as Editor's Choice in 2007, and also in Current Science under Research News in 2008. So then, as I mentioned earlier, Monocillin I, we needed large quantities, but the fungus produces only small quantities of this compound. But fortunately for us, at that time, what happened was, so we tried under a variety of conditions, and we got only very little amount of Monocillin I. That is the peak corresponding to Monocillin I, very little amount. Then that was a time I had a scholar from Sri Lanka. I forgot to tell her that we should use distilled water to make up the fungal cultures. By mistake, she used tap water. And in that, you got large amount of Monocillin I. So it was accidental discovery. Good that I didn't give her those instructions, right? Sometimes these things happen. So now I do that. I don't give all the instructions. Let them make a mistake to see whether we will make another chance discovery. So we got large quantity of that, and we were able to. That's my, Fulbright scholar who came from Sri Lanka, and so then what we did was we analyzed tap water, which we used, and we found that tap water in Tucson at that time contained unusually high amount of copper. Then we were able to use copper, 0.25 millimolar copper sulfate, and we were able to produce, in one run, we were able to produce about 1 gram of this rare compound, and we were able to give it to Valent Biosciences for their studies. Okay, so what are the implications? This is probably far-reaching, but as I say here, much more work needed to be done before it becomes a hit. Now, we provided proof of concept. That's all we can do as a natural product chemist. It's up to plant scientists to pick it up. And as I mentioned earlier, can it be an alternative to genetic modification? Can it avoid water shortage? Because water is also a stress, like thermal stress. Can it help reduce desertification? So these are the questions which we have to answer later on. Okay, these are microorganisms which Rafi worked on this plant in our greenhouses when we were there. We were growing some plants aeroponically, not hydroponically, aeroponically. We were growing, I'll talk about that in a minute. And this is one of the plants called bladder cherry, Physalis alkatengii, and we isolated, we investigated many plants to see whether they harbor any endophytes. And this was the only endophyte we were able to find in environmental controlled plants. Whereas if you go to the field, you will find hundreds of them, hundreds of microorganisms. Maybe under environmental controlled conditions, they don't need microorganisms to survive. That may be the reason. Plants are very smart. That may be the reason why they don't harbor. And we isolated several new compounds with new carbon skeleton, and that was grown. So after that experience, we always add copper to our medium, right? We always add a little bit of copper, and we have found that it actually works. It produces more metabolites, and it produces the extract quantities much higher than without copper. So this was published in 2014. You can see that Rafi was involved in that work. Okay, now, are there any questions? Before I move on to plants. So that's about microorganisms. Let me quickly move on to plants, and I will stop when it comes very exciting. So we have about 5,000 desert plant extracts and over 13,000 extracts. So what we do is, in the case of both plants and microorganisms, we have put them into 96 well plates. And we provide these to our collaborators without telling them what they are. So they will do the bioassay, and they will tell us, Leslie, plate number 1, well number 6 was active. So we get the information. Then we contact our tech transfer office. We sign agreements, and under confidentiality agreement, we give them, we tell them what it is. Then we start our work to discover the compounds. So this is what we did. And one of the plants which is native to Sonoran Desert is called Physalis crassifolia. And from that plant, we isolated a compound which belongs to the class of withanolides. And it was active. It was not active in heat shock induction assay, but it was active only in cytotoxicity assay. And interestingly and excitingly, it showed selectivity only to prostate cancer cells, very high selectivity. It didn't kill any other cell line we have tested. We have sent it to NCI, we have sent it abroad to England to test, no other activity, only prostate cancer cell lines. So, and then we wanted to use that to develop a lead molecule, use that as a lead molecule to develop drugs for prostate cancer. And the second plant which I'm going to talk about is a well-known plant, Withania somnifera. It is found in desert regions in Asia, and it is used, that I mentioned, used as a medicinal plant in Ayurvedic medicine. And the compound which we isolated, it's a known compound called withanolides A. And this was active in both heat shock induction and cytotoxicity assays. So it is good for neurodegenerative diseases and also for cancer. So we want to see whether we can develop it as a drug to treat neurodegenerative disorders and also for cancer, right? So we have done some chemistry with regard to that. So these are some beautiful plants, all belonging to the genus Physalis crassifolia. Look at that. It's a sad-looking plant which grows in Sonoran Desert, right? It's very hard to collect, and it's very hard, very hard to find the plant. It's called Physalis crassifolia, yellow nightshade ground cherry. Then alkatengi is the one from which we isolated that endophyte. It's called Chinese lantern. And then this is Physalis peruviana, golden berry. We have studied that as well, and also Physalis minima. Now, how they describe this plant is it's a curious green to orange fruits wrapped in a lantern-like husk. The husk is like a lantern. So that is why this is called Chinese lantern. The plant, the flower or the fruit is called Chinese lantern. These are used in China, India, and also in North America. It is used in India to treat cancer. This one, Physalis minima. It is called native gooseberry. So what we did was, all these 18,000 samples we assayed in our cytotoxicity assay, And we found, as I mentioned earlier, this was active only against prostate cancer, and we isolated 4 compounds. You see, LN cap is the hormone-sensitive prostate cancer. It was active at 40 nanomolar compared to normal cells, HFF, that is human foreskin fibrillar cells. It was active, it was not active up to 3,140 micromolar, right? So here it's 40 nanomolar. And then second compound was also active, not as active as this one. Then they were also active against PC3, that is a hormone-insensitive prostate cancer. And this is the metastatic version of hormone-insensitive prostate cancer, again, much more active than the normal against the normal cells. So we were really excited about that. And then we collaborated with Nuogen Research. That's a company small company based in Tucson. And they subjected our 18,000 extracts to a new assay, which is called the array plate gene expression assay. They were looking for expression of genes related to prostate cancer. And our compounds were found to inhibit prostate PSA, right, at 270 nanomolar. So we are trying to develop this as a drug now in collaboration with the National Cancer Institute. So this is the problem with all these, as I mentioned earlier, is the supply. Plant is hard to grow. Plant is hard to find. Even when you find it, it looks very sad, right? It's a very small plant, not very flourishing type of plant. We needed this compound for assays, so it is not commercially available. So we had to, we tried a method which we have developed earlier for Withania somnifera, which is called the aeroponic technique. Here what we do is we hang the plant, we hang the seedling in a box, and we spray the root system with a nutrient medium. And the plant grows very happily, matures much faster than when it is cultivated in soil, and then we have large amount of biomass. Very quickly, we isolate our compound. So by this method, we were able to do some aeroponic boxes. So what we have done was we have taken hydroponic boxes and modified them, right? We have put just a wooden box around it, and it's modified, and every 10 minutes, the nutrient medium is sprayed to the root system for 1 minute. And if you want to change, if you want to stress the plant, we can stress it. We can spray it every 15 minutes instead of 10 minutes. Or if you want for it to get more nutrients, if it's growing slowly, then we can spray it every 5 minutes. So that is the difference between aeroponics and hydroponics. Hydroponics, you provide the nutrient all the time. But aeroponics, you provide the nutrient, at a given time for a certain period of time, and then you can stress the plant. Stressing is important. Now, biomass is important when you want to use it as a food. Stressing is important when you want to isolate secondary metabolites, because plants produce these compounds when they are under stress. So we can experiment. And also, I read somewhere that this technique uses only 10% of water, which is required when you grow the plant in soil. So here, by using this method, we were able to produce from 12 aeroponic chambers 12 grams of this compound, right, for experiments. So having enough compound, we were able to do some studies. Since you will recognize that this has a steroid-like structure, so we wanted to eliminate the possibility that it affects the androgen receptor. Because if it affects the androgen receptor, then it is of no use, because it acts as a steroid. So the first experiment we did was to show that it doesn't act as an androgen receptor agonist. And this was actually, we didn't have access to the screen. We signed agreements with Invitrogen company, and they did this assay for us. Of course, we had to pay for that. And they showed that it does not bind to the androgen receptor. This is actually dihydrotestosterone, which binds to the androgen receptor. this is our compound, no binding at all. So we were, again, very pleased about that. And then we determined toxicity using animals. My collaborators did that at the Cancer Center. And it was found that the compound was not toxic up to 50 milligrams per kilogram. Now we are ready to do efficacy studies. We did efficacy studies and found that at the concentration, dose at which it was not active, 50 milligrams per kilogram, it reduced the tumor mass of PC, this is PC3, it reduced the PC3M xenografts, this is the metastatic version, reduced by about 50%. So we are very excited. We did more experiments. The problem during animal studies was that the compound was found to be unstable. So that is where chemistry knowledge is important. We converted that into a stable compound. So this was found to be unstable because this position is very unstable in solution. So we converted that into epoxide. We got about 50% yield, and that was found to be even more active. 2 was even more active. It was active at 22 nanomolar instead of 75 nanomolar. And then it was also active in our, the neogen gene expression inhibition assay. So this is the compound which we are trying to pursue now. We have made the compound stable, and then we are trying to develop that as a drug. So these are, more animal experiments. This is PC3 xenograph. This is LN cat. So we have used all 3 mouse models to show that our compound is effective, but there is no synergistic effect with Taxol. We tried treating with Taxol. Giving them together, there was no effect. But our compound was active as it is, and Taxol was active as it is. There is no synergism. Okay, so this was published in 2015, and then we grew the plant. When we grow the plant hydroponically, we found that there are so many new compounds produced. They're not produced by the plant when it is cultivated in soil. So this is what you call, diversification of structures. We have diversified the structural diversification. We have got so many compounds. And having those compounds, for which this was published, Anura was involved in this work. He was a volunteer visiting scientist in my lab from Prima Community College, and we produced large number of compounds, and they also showed selective cytotoxicity to prostate cancer cells. So having so many compounds, we were able to find out which part of the molecule is important for activity and so on. Okay, so move on to Withania somnifera. We applied the same technique. It's a plant which takes so many years to mature, 2 to 3 years to mature. When we grow aeroponically, it reaches maturity, produces fruits in 6 to 8 months, and you see the comparison of the leaf of a plant grown in the same greenhouse, in soil and under aeroponic conditions. The biomass is about 5 times. And then we were able to isolate 2 compounds actually. This is a prodrug of withanolides. It is very important. Prodrugs are drugs which are just before the drug. They are water soluble, and then when you give it to an animal or human being, it gets converted into the drug. So the water solubility for a drug is very important. What pharmaceutical companies do is, when they find a drug, they try to convert into a water-soluble analog, so that instead of injecting it, you can give it orally if it survives that. And then we were able to produce about more than 100 grams of this compound. I checked the price today from Sigma Aldrich. 5 milligrams is $367. So if you want to calculate, you can calculate, if you have a calculator, how much this 100 grams is worth. We have not sold a single milligram to anybody. We have given it for collaboration. And we have a patent for that, finding production of withanolides A using that method. We have given to so many collaborators, right? I'm going to talk only about few collaborators where I have the smiley face. And this was a very, very sad story. They took our compounds and they applied for their, this is a company, they applied for their own patent, right? So I'm sure there'll be a lawsuit very soon, right? They applied for a patent. This is for ALS. They want to develop our withanolide analogs to treat ALS, Lou Gehrig's disease, right? I'm having very interesting collaboration with National Cancer Institute. And here, what the National Cancer Institute is interested in now is they have looked at many targets. So they are trying to, they have a program, they have developed a program to discover anti-cancer drugs which act by novel mechanisms, new mechanisms. And they were looking for what you call trail sensitizing agents to induce apoptosis. Now, apoptosis is very important because these are 6 hallmarks of cancer. Cancer cells have learned how to evade these, 6 hallmarks. That is why they are different from normal cells. So one of them is evading apoptosis. Apoptosis is committing suicide. Normal cells, when they are stressed too much, they commit suicide. But cancer cells, they don't commit suicide. It's very hard for us to get them to commit suicide. So this is what, if you look at TRAIL, stands for tumor necrosis factor alpha related apoptosis inducing ligand. And this is the ligand which induces apoptosis in cancer cells. So what happens, unfortunately, is when you treat cancers with anti-apoptotic drugs, cancers are very smart. They develop resistance. So therefore, you have to sensitize TRAIL. This ligand, you have to sensitize. That is why NCI is interested in looking for these sensitizers to induce apoptosis, induce committing suicide of cancer cells. So what they did was, they took 50,000. We used only 18,000. They used 50,000 natural products and synthetic compounds. And they screened, and lo and behold, what they found was similar to our compounds. And that was the most promising compound, was withanolide E. So they contacted me because they have seen our publication. They contacted me. I went there and gave a seminar. They were very much excited about our compounds, and they want to test our compounds. And then we provided, at that time we had 98. Now we have about 150. 98 withanolides from our library. These are natural and semi-synthetic withanolides. We provided these to them, and they found this particular compound, which we found to be active in prostate cancer assay, to be about 4 times more active than their compound. And this is a synthetic compound, which we can patent. Natural products you can't patent, but synthetic compounds, synthetic analogs, you can patent, and which can be patented. And then this is about 8-fold more active than withanolide E for melanomas. You know that melanoma is a big problem in Arizona. It's a type of skin cancer. And it is very interesting that these compounds are active against melanoma. And then we want to see whether they can be used as immunotherapeutic drugs. Now in 2013, Science magazine announced this as the cancer immunotherapy as the breakthrough of the year, cancer immunotherapy. And also, I'm sure you have heard about Cancer Moonshot initiative by Vice President Joe Biden, right? And they think immunotherapy, cancer immunotherapy, is the answer to most of the cancers which are resistant to drug therapy. So I asked my collaborator to test whether any of our compounds is active in the assay. So here we have anti-cancer drugs. What we do is you combine the drug with immune adjuvants, and then there are number of clinically effective combination immunotherapeutic drugs known. For example, the one which our former president, right, he's using for melanoma is actually a combination drug, immunotherapy drugs. It's a drug plus immune adjuvant, and he's doing quite well with that. So when we tested, what we did was we tested this compound, our phycocyanide C, which we got from Physalis crassifolia, which we cultivated under aeroponic conditions, and then used immune adjuvant, which is a mismatched double-stranded RNA. It's called poly-IC. And then we did animal studies, injected animals with melanoma, and then we used a set of animals. We used only the vehicle. We used only the compound, only poly-IC, which is the immune adjuvant, and a combination. And then what we see is that this is the vehicle. Tumor grows in animals. This is poly-IC alone. That is the immune adjuvant alone. And this is our compound alone. And when you combine, cancer is completely gone. So these are very exciting results, which I told Rafi about. We have not published this work yet. And this is just the beginning. So we are now collaborating with Dr. Thomas Sayers of NCI, he's a cancer immunologist, to see whether we can discover better drugs to treat not only melanoma, but also other cancers, renal carcinoma and so on, because he's a specialist in renal carcinoma. So that is the one which I want to draw your attention to. Do you have time, or should I stop? I can go ahead. Great, good. So we have a patent application. There is a patent application for this provisional patent, and also NCI has already advertised. NCI Technology Transfer Center has advertised the availability of this for licensing and also collaboration. So companies look at these things, and if they are interested, they will contact us. So this is a provisional patent by NCI and University of Arizona. So University of Arizona will also benefit if we can find immunotherapeutic agent. Okay, so let's move on to our favorite plant, Withania somnifera, ashwagandha. It's also called Indian ginseng. And here, this is the plant where we saw activity in our both assays, cytotoxicity assay and heat shock induction assay. So we wanted to modulate the structures, sort of change the structural scaffold to see whether we can get one class of compounds which are active against cancer, the other class active against Alzheimer's and Parkinson's. Of course, for that, you have to do chemistry. And then what we did was, so this is about the plant. It has been in use for over 3,000 years in India, right? It is called ashwagandha, and it's called somnifera. Somnifera is to sleep, right, in Latin. So it is a nerve sedative. So it should have some activity towards your nervous system. It is also used as an immunomodulant, like ginseng, Chinese ginseng. So it got its name as Indian ginseng. And it is available over the counter. We have a bottle here, right? And the latest one, I don't know how many of you have heard about this. This is called Protandim. Only yesterday I bought a bottle. There's only one supplier, and she is a private person. I had my class yesterday. I want to show it to my students. So I got her to bring it to campus. I paid $50 and bought this. It's $60. She gave me a discount because I was going to show it to my students, right? And this is very interesting. It's a combination therapy. It has a lot of antioxidants. You must go to the website and see what they claim, right? They say you can remain like a child forever, right? Anti-aging. It stops aging. Oxidative stress. Because oxidative stress is a big problem. Not only it helps you to age, right? It also is responsible for many diseases. heart diseases, diabetes, cancer, and so on. So this contains ashwagandha, it contains green tea, decaffeinated tea, it contains several other plants which are known to have antioxidant activity. Right? So this is the one. And it is also sold as mind power. Right? My students like it. Mind power. And then also as ashwagandha. So it's actually the root which is used. Yeah, this part which is used as a drug. So we subjected, this 18,000. This is one of the plants we found to be, or one of the extracts, which was found to be active in our heat shock induction assay. We did a bioactivity guided fractionation. These are the two compounds which we isolated. This we published in 2009. And then it was found to be active against a number of cell lines, but it had some moderate selectivity towards nervous system associated cancer cell lines, right? Like glioma and neuroblastoma. So we, in collaboration with the Whitehead Institute at MIT, we tested this against brain tumor, and it was found to be better than the currently used drugs, right, for brain tumor. I'll talk about that in a minute. So we wanted to do further studies, and we were able to produce, as I showed you, aeroponically, we were able to produce large quantities, and then we were able to identify the protein target, which was published in Nature Chemical Biology in 2006. Then we tested against pancreatic cancer. It was found to be active, published in 2010. See the activity here is a toxicity, it's active. And then brain tumor with MIT, collaboration with MIT, which was published in ACS Chemical Biology in 2012. It's found to be active, reduced brain tumor mass in animals. These are all animal studies, not human, please. And then we prepared more analogs, and in collaboration with the Cancer Center, we again showed that some of the analogs were more active towards pancreatic cancer. And then recently we showed, that's 2015, we showed that this analog is a brand new analog with nitrogen at these places, which we synthesized, and that was active against AAA chaperone, which is again a very important protein target for cancer and other diseases, even diabetes. Okay, so we were able to do additional structure-active relationships. This is the outcome. This is what we did. We not only do chemistry, we also get microorganisms to do biological transformations. Because, for example, a chemist will take ages, generations, to introduce a hydroxy group here or hydroxy group there into this molecule. We get microorganisms to do it very efficiently. So in our lab, we even make use of microorganisms to convert natural products into useful analogs. And then, remember I mentioned the connection between heat shock proteins and neurodegenerative diseases. Now we have shown, up to now we have shown that withanolide analogs can be used as anti-cancer drugs. So we wanted to see whether they can be developed as drugs for neurodegenerative disorders. So here we need to actually reduce the cytotoxicity and increase heat shock induction activity. For cancer, we have to reduce heat shock induction activity, increase cytotoxicity. So it is opposite. So we want to do what we call cytoprotection. And so this is it is actually shifting the activity spectrum from one end to the other end. And we were able to do that, right, to increase heat shock induction activity so that we can use it as neurodegenerative agents, anti-neurodegenerative agents. And here we have already seen that they have anti-cancer activity. So we have already synthesized about 36 analogs plus withaferin A, 38. We tested them in heat shock induction assay, and we found that is where withaferin A our parent compound, lyse, but there were several other compounds which showed higher heat shock induction activity than withanolides A. And these were all our analogs. These 5 analogs showed higher activity. And then these 2 actually, these are synthetic molecule, these 2 showed higher activity. And then when we plotted cytotoxicity against heat shock induction activity, there was a cluster of compounds where the heat shock induction activity was high compared to cytotoxic activity. So therefore, we wanted to test, especially this compound. Well, that's a compound which you can patent. It's a new compound, it's a synthetic compound, derived from withanolides A. So we tested this one to see whether they have activity in animals, and we used heat shock induction. We fed into mice, and then we took the spleen. This is done by my collaborators. I said I can't do animal studies, but my collaborators can. And they found at a dose of 25 milligrams per kilogram, there was induction of HSP72 in spleens of those animals, given our analog number 35 compared to withanolides A. And then at 40 milligrams per kilogram, it was even higher. So this work was published in JMedChem, Journal of Medicinal Chemistry in 2015. And then a startup company called Humanity Therapeutics are looking for neurodegenerative agents or drugs to treat neurodegenerative diseases, right? And we were able to license our patent to them. So now they are trying to use these analogs to prepare anti-neurodegenerative drugs. So just before I talk about some breaking news, which I got just before I prepared for this presentation, this is just a summary and highlights. We have discovered a compound natural product from a fungus, plant-associated fungus, which is capable of imparting or conferring thermotolerance to Arabidopsis. And then we have discovered a class of withanolides from desert plants with heat shock induction activity and also cytotoxicity, some with selectivity with prostate cancer. And then we have demonstrated their in vivo activity. We have applied an aeroponic technique to cultivate withanolide producing compounds, and we have been able to produce large quantities of withanolides. And then we have discovered combinatorial immunotherapy to treat melanomas, and then we have demonstrated that the withanolide scaffold structure can be manipulated if you want to get cytotoxic drugs to treat cancer or drugs to treat neurodegenerative diseases. So I would like to thank this is just some of my collaborators who have contributed to this program. These are my co-workers. You see Rafi's name there, Hema's name there, Andru Bandarnayaka. These are the people who directly participated in the program. These are my visiting scholars, mainly from Brazil, who participated in the project. These are my collaborators from University of Arizona, from Federal University of Sierra, that's in Fortaleza, Brazil. Dr. Eli Chapman from College of Pharmacy. Susan Lindquist, she's a very well-known heat shock protein specialist. Actually, she got President Obama's medal for science last year. Susan Lindquist, and then Catherine from Harvard, and Luke Whitesell is my main contact at Whitehead Institute, and these are the funding agencies. I'm just a spokesman for all these people funded by these organizations, and these are the people who really worked hard. So I would like to thank all of them. I would like to thank all of you for your attention, and here is the breaking news. It appeared in this week's Nature Medicine. Withanolides A is a leptin sensitizer with strong anti-diabetic properties in mice. And news and views, obesity will withanolides A win the war. So our ability to produce large, we are excited because our ability to produce large quantities of withanolides A by our patent technology, I think if they want to use it for human studies, we can be the supplier and the university will benefit from that. So with that, I would like to stop my presentation.
Thank you very much. Thank you very much. All I can say is plants rock. And we have seen a fantastic example, in my opinion, of the marriage of agriculture, plant growing, chemistry, and most importantly, the enthusiasm of human creativity. So thank you very much. And we have a few minutes for some questions. If anybody wants to volunteer to be the first one. After that, it's much easier. Merle, are you going to go? Okay. That's a big compliment.
Welcome.
Okay. Well, I guess there are no questions, so... Oh, okay, question. There we go.
I would have been very unhappy if there were no questions.
I was wondering when you said when you grew the plants aeroponically, you got many more compounds than when you grew them in soil. And had you grown them in soil where you were able to stress them? Is it a matter of being able to stress the plant, or why do you think
we grew them under the same conditions? But of course, they were getting more nutrients, actually, than the aeroponically grown plants. So we believe that structural diversification may be due to stressing. Okay? And also one thing I forgot to mention, which is very important, is that when you grow Withannia somnifera in soil, withanolides A is found in roots. When you grow it aeroponically, withanolides A is found in shoots, in aerial parts, which we can continuously harvest, and also it is present as a water-soluble compound. So what I believe is that it is produced in large quantities by the plant, and it is water-soluble now, and it is non-toxic. Whenever the plant needs it for a particular purpose, for example, to repel insects or whatever the feedant is of the plant, then it will release the compound. Oh, good.
One of the things that for stress in aeroponics, have you tried to also increase along with that the electrical conductivity like the soil?
No,
You have not done that.
We have not done. Only thing we have done was to incorporate well-known stressors like salicylic acid elicitors, like salicylic acid and plant hormones. There's a little bit of increase of natural product production by the plants when you use salicylic acid, but it's not significant.
Yeah, we can stress tomato plants, for example.
Mm-hmm. Yeah. Thank you. Thank you for keeping for so long. Thank you for waiting. Sorry for keeping for so long. Soon. Yeah. Well, humanity, they are really interested.